All right, let's just jump right in. I want you to think about something. Right now, as you're listening to me, there's this microscopic dance, an unbelievably complex one, happening inside you trillions of times over. This is the process that literally built you from just one single cell, and it's still going, repairing and maintaining you every single second. Yep, we're talking about cell division.
Seriously, just let that number sink in for a second. 2 trillion. That breaks down to over 20 million new cells every single second. Now, the big challenge, the real problem your body has to solve, is making sure that every single one of those new cells gets a perfect, flawless copy of your genetic blueprint. So how in the world does it pull off such an incredible feat?
Okay, so to really get how cells divide, we've first got to look at how they store all their instructions. You can think of it like this. Every single one of your cells has a complete instruction manual for building you, and it's all packed away super neatly in what we can call a genetic library.
And this is where it gets kind of mind-bending. If you took the DNA from just one of your cells, just one, and you unspooled it all, it would stretch out to be about 6 feet long. So the puzzle is, how on earth do you cram a 6-foot-long manual into a cell nucleus that's completely microscopic?
Well, the answer is just, it's a masterpiece of packaging. I mean, it's brilliant. That long string of DNA gets wrapped around these special proteins, then it gets coiled up, and then coiled up again until it's packed down into these super dense structures called chromosomes. So if the DNA is the library, then each chromosome is like a single book, a separate volume on the shelf.
Okay, this leads us to a really important idea. You see, most of the cells in your body, think skin cells, muscle cells, all that stuff, they're what we call diploid. That just means they have 2 full sets of chromosomes, one set you got from your mom and one from your dad. But your reproductive cells, so sperm and egg cells, they're different. They're haploid, which means they only have one single set.
So why does that even matter, right? Well, it's the whole point of sexual reproduction. You take one haploid cell, the sperm, and another haploid cell, the egg, and you combine them. That process, which we all know as fertilization, creates a brand new diploid cell. We call it a zygote. And that little cell has the complete, full set of chromosomes needed to grow into a whole new person. All right, now that we've got that background, let's get into the first big dance of division. It's called mitosis. This is the workhorse process your body uses for just about everything, for growing bigger and for repairing itself, like when a paper cut heals up.
The goal here is super simple, but really elegant. Mitosis is basically the cell's photocopier. That's its whole job. It takes one cell, the parent cell, and makes 2 new daughter cells that are perfect, identical copies. No shuffling, no changes, just a straight-up duplicate.
Now, this whole thing happens as part of what we call the cell cycle. And what's interesting is that the cell actually spends most of its life, you can see it in that big blue section, in a phase called interphase. This is all the prep work. It's growing, it's doing its job, and most importantly, it's copying all of its DNA. The actual division part, mitosis, is a really short and, frankly, super dramatic finale to the whole cycle. So mitosis itself breaks down into 4 main stages. There's a handy little acronym that biologists use to remember them, PMAT. That stands for prophase, metaphase, anaphase, and telophase. Let's do a quick walkthrough of what's happening in each step.
All right, first up is prophase. Here, those copied chromosomes start to thicken up and condense so you can actually see them, kind of like thin threads bunching up into thick ropes. Then comes metaphase, and this is where they do something really cool. They all line up perfectly, right down the middle of the cell. It's like dancers getting into formation for the main event.
And then we get to the most critical step of all, anaphase. This is the moment of truth. Tiny little fibers reach out, grab onto those lined-up chromosomes, and yank their identical halves apart. These halves are called sister chromatids. One complete set of instructions gets pulled to one side of the cell, and the other identical set gets pulled to the opposite side. This is the move that guarantees each new cell gets a perfect, complete set of books from the genetic library.
So after the chromosomes make it to their new homes in the final stage, telophase, the cell has one last job to do. It has to split its main body, the cytoplasm, in 2. This final split is called cytokinesis. And here's a fun little fact. Animal and plant cells do this differently. Animal cells kind of pinch in the middle like tying a balloon, but plant cells have to build a brand new wall right down the center to separate the 2 new cells. Okay, so that's mitosis. That's how we make perfect copies for growing and healing. But what about making babies? What about creating the genetic variety that's so important for reproduction? Well, that calls for a totally different dance of division. It's called meiosis. And if mitosis is a photocopier, then meiosis is like a MasterCard shuffler. Its goal isn't to make perfect copies. Its goal is to create diversity.
Meiosis has two huge jobs to do. Job number one, it has to cut the number of chromosomes in half. It needs to go from a diploid cell with 2 sets down to those haploid gametes with just 1 set. Job number two, it has to shuffle up the genes from the parent's original chromosome sets. This is what makes sure that every single sperm or egg cell is one of a kind, genetically unique.
So how does it shuffle the deck? The first way is through this amazing process called crossing over. What happens is, early on in meiosis, the chromosome you inherited from your mom gets real close to the one from your dad, and they literally trade pieces. They swap sections of DNA back and forth. This creates brand new, never-before-seen combinations of genes on those chromosomes. And that's not all. The second shuffling mechanism is something called independent assortment. When all those chromosome pairs line up in the middle of the cell, how they line up, which one's on the left, which one's on the right, is completely random. It's a total crapshoot. And this process alone creates a staggering number of different combinations. Just in humans, this one step can generate over 8 million possible combinations in the final cells. Insane, right?
Now, with all this complex choreography going on, you've got to be thinking, things must go wrong sometimes, right? So how does the cell keep everything in order? How does it stop things from going completely off the rails? Well, it turns out it has a really sophisticated quality control system. Throughout the cell cycle, there are these things called checkpoints. You can think of them like super strict quality control inspectors on an assembly line. There's one that checks for DNA damage before copying starts. There's another that makes sure the DNA was copied perfectly. And there's even one that double-checks that all the chromosomes are lined up correctly before they get pulled apart. A cell literally cannot move on to the next step until it gets the all clear from each inspector. It's a nearly flawless system. But it does raise a pretty scary question. What happens when the inspectors are asleep on the job? What happens when a bad cell, a damaged cell, slips past a checkpoint and gets the green light to divide anyway? Well, the answer is something we are all unfortunately familiar with: cancer. When you get right down to it, cancer is a disease of the cell cycle. It's exactly what happens when these critical checkpoints fail. Damaged cells start multiplying, dividing over and over, completely ignoring the body's stop signs. So there you have it. We've looked at two very different but equally amazing cellular dances, mitosis and meiosis. And while they seem totally different on the surface, they both serve the same ultimate purpose of all life: to keep going. So let's just put them side by side one last time to make it all crystal clear.
Yeah, this really sums it all up, doesn't it? For mitosis, the purpose is growth and repair. It does one division, and you end up with 2 identical diploid cells. For meiosis, the purpose is sexual reproduction. It goes through 2 rounds of division and ends up with 4 genetically unique haploid cells. So you can really think of it this way. One process is all about continuity, and the other is all about creating variety.
And that's the thing. In the end, these incredibly intricate molecular dances are happening inside you right now with almost perfect precision. But the fact that even tiny, tiny mistakes in this choreography can lead to something as devastating as cancer, well, that tells us something really profound. It shows us the incredible tightrope that life itself is always walking, a delicate, beautiful balance between perfect precision and random error, between incredible resilience and heartbreaking fragility.
Subscribe to my channel.